So think about your driver's license for a second. It gives you an exact number, right?
Yeah.
Your chronological age.
Right.
But if you actually look around, you've got, some 70-year-olds who are out there running marathons, and then others who are unfortunately bedridden.
Yeah. It's a massive difference.
Exactly. And that's your physical state, your biological age. Today's deep dive is really about digging into that gap. We're looking at this revolutionary scientific paper introducing something called scAgeClock.
Which is just honestly a total paradigm shift.
It really is. It's an AI model, but it doesn't just look at the age of your body as a whole. It effectively acts as a biological clock for your individual cells.
Which is the crazy part, because we're so used to thinking about aging as this uniform thing, like time passes and the whole machine just slowly degrades at the same rate.
But this study shows that's just not true.
Right, not at all. Your body is basically this chaotic mosaic of different biological time zones.
Time zones, I love that.
Yeah. And early aging clocks, they couldn't see that. They were kind of like looking at a crowded stadium from a blip. You're just averaging out the entire crowd.
Right.
But scAgeClock uses single-cell RNA sequencing, so it looks at the exact specific transcriptomic blueprint of individual cells.
And the scale of this is just wild. I mean, we're talking about a data set of over 16 million individual cells.
16 million.
From more than 40 different human tissues.
Yeah.
400 distinct cell types. And the donors ranged from like 1-month-old infants all the way to 96-year-olds.
It's massive. And to really understand why this is such a breakthrough, we kind of need to look at the problem at the old way we used to measure biological age.
Moving past the smoothie, basically.
Right, exactly. The smoothie problem. So previous aging clocks mostly relied on bulk level samples. You'd take a vial of blood, for example, and look at DNA methylation.
Okay, so let's unpack that. With bulk sequencing, it's like, if you use an analogy here, it's like throwing a bunch of strawberries, some blueberries, maybe some bananas into a blender, right? You hit blend, you taste the resulting smoothie, and then from that one sip, you try to guess the exact age and ripeness of just the strawberries.
Which is basically impossible.
Right, because you're grinding together immune cells, structural cells, blood vessel cells, all into one average gene expression profile.
And that average is super deceptive. Biologists call this the heterogeneity problem.
Heterogeneity meaning just the mixed up nature of the sample.
Exactly, the mix. So let's say you have a viral infection in a specific tissue. You run a bulk sequencing test before the infection and then another one after.
Okay.
The data might show that the tissue suddenly aged, like 10 years.
Wow, just from the infection.
Well, that's the thing. The individual resident cells didn't necessarily age at all.
Oh, wait, what?
Yeah. What happened was the infection triggered a massive influx of immune cells, macrophages, T cells rushing to the area.
Ah, so the recipe of the smoothie changed.
Exactly. The ratio of the different cell types changed, so the whole sample looks different. And without single-cell resolution, scientists could draw completely wrong conclusions. They'd think, oh, the cells aged faster, when really they're just new cells in the mix.
So single-cell RNA sequencing, or cRNA-seq, is like pulling out every single strawberry, every blueberry, and just inspecting them individually.
Yes, you're looking at their individual transcriptomic to-do lists.
Okay, so DNA is the reference library, but RNA is the active, real-time to-do list for the cell.
Perfect way to put it. It tells us exactly which proteins the cell is trying to build at that exact second.
But inspecting 16 million of these individual to-do lists? I mean, a human can't do that.
No, not a chance. You need a highly specific kind of artificial intelligence to make sense of it all.
Right. And this brings us to the brains of the clock. The researchers used an architecture called a gated multi-head attention neural network, or GMA.
Right. And just to give some context, they tested this against some heavy-hitting baseline AI models.
Like XGBoost, CatBoost.
Yeah, and standard multi-layer perceptrons, or MLPs. And the GMA model just completely outperformed them. The error rates were incredibly low.
Okay, let's unpack this for the listener, because multi-head attention is a dense term. What exactly does that mean when we're reading cellular data?
So a standard neural network can process a lot of information, but it kind of struggles to weigh the contextual importance of different data points against each other.
Right.
But attention in AI is a mechanism that lets the model dynamically focus on specific parts of the data that are most relevant to whatever task it's doing.
Okay, so let's think about it like investigating a massive crime scene.
I like that.
Right, so standard model is like one detective just trying to look at everything in a vacuum, a footprint, a bank statement, whatever.
They're overwhelmed.
Yeah. But multi-head attention is like sending in a specialized team. You have one detective who only looks for fingerprints, another one only checks financial records, another interviews witnesses.
And then they work in parallel to synthesize the big picture.
Yes, exactly. The fingerprint detective realizes their clue is only important if the financial detective finds a matching bank withdrawal. They weigh their clues against each other.
And in biology, that relational weighting is everything. Because genetic pathways interact, the AI isn't just looking at 19,000 genes in isolation, it's seeing how gene A changes the context of gene B.
But there's a problem, right? The single-cell data is really messy.
Well, it's notoriously noisy.
Yeah.
Because you're dealing with microscopic amounts of RNA inside just one tiny cell.
So the sequencing machine sometimes just misses things.
Yeah, we call those technical dropouts. They register as false zeros in the data. So your AI detectives are constantly being bombarded by these false clues and irrelevant genetic noise.
Which is where the gated part of gated multi-head attention comes in. The feature gate.
Right, the captain of the detective squad, basically.
And he uses a mathematical penalty system, right? L1 and L2 regularization?
Exactly. Think of the feature gate like a ruthless editor reviewing a massive essay. If a specific gene's expression isn't actively contributing to predicting the cell's age, this regularization algorithm mathematically squashes its influence down to zero.
It just cuts it out.
It forcibly silences the irrelevant noise. That way, the attention heads, the specialized detectives, only focus on the strongest, most important biological pathways.
And without that gate, the model falls apart.
Yeah, when they experimentally removed the feature gate, the accuracy just plummeted. The AI needs that strict editor.
Wow. Okay, so before we jump into what happens when this biological clock actually breaks down, let's do a quick mid-dive recap for you listening just to make sure we have the foundation set.
Sounds good.
Okay, bullet 1. Biological age differs from chronological age. Bullet 2. Single-cell RNA sequencing lets us look at the individual cell level, separating the smoothie. And bullet 3. The GMA neural network is the parallel processing brain reading all the cellular data.
Perfect summary.
So let's get into the actual biology. The AI looked at 19,234 protein coding genes. What did it actually learn about human aging? What were the top features?
Well, it went way beyond just simple wear and tear. One of the top features it flagged was ribosome genes.
Ribosomes. So those are the factories making the proteins.
Right. They handle translation. If your ribosomes get inefficient or start making errors over time, you get a buildup of misfolded proteins. And eventually, cellular function just collapses.
Makes sense. What else?
It also flagged genes linked to the viral life cycle and to programmed cell death, which is called apoptosis.
But it also found some genes we already know are huge in longevity science, right? Like APOE.
Yes, APOE was heavily weighted. We usually hear about it with Alzheimer's risk, but the reason it's a reliable aging clock across all cells is because it manages lipid transport.
It's the garbage disposal.
Exactly. It cleans up cellular debris. As that system breaks down, the trash builds up. The AI mathematically spotted that cleanup system failing.
And it also flagged LMNA, which is fascinating because that's the progeria gene.
Right. LMNA handles the structural integrity of the cell nucleus. When it mutates, it causes progeria, which is that tragic disease where children experience rapid, accelerated aging.
So the AI figured out on its own that the slow degradation of that same gene is a universal marker for normal aging in everyone.
Completely independently, yeah. But here's the crazy part. The biggest surprise wasn't about things breaking down.
Right, the peak. This blew my mind. Out of the top 20 aging genes, 15 of them don't just slowly fade away. They actually peak in expression around age 30.
It completely flips how we think about aging. We usually think, from the day you're born, you're just slowly degrading.
A slow, depressing slide.
Yeah. But the transcriptome shows it's actually a climb. You're building and optimizing these genetic pathways until your cellular architecture hits its absolute zenith at 30.
And then the winding down begins.
Exactly. Only then.
And different cells age differently during that wind down. The model found that blood and colon cells are pretty easy to predict.
Yeah, they have a steady cadence.
But skin, liver, and pancreas cells, massive variability. Why is that?
Environment. Your skin is a barrier. It's getting hit by UV radiation, pollution. Your liver is filtering toxins and managing metabolic stress based on what you eat and drink.
So the cells are constantly repairing and turning over.
Right, which means their biological age can diverge wildly from your calendar age.
So if our bodies are this chaotic landscape, how do we actually measure if a cell is aging too fast? Like, how do we put a hard mathematical number on that chaos.
Enter the ADI, the Aging Deviation Index.
Okay, let's explain this scale. It maps the age deviation from 0 to 1.
Yep, and the magic number is exactly 0.5.
Okay.
If a cell has an ADI of 0.5, it means the AI's predicted age matches the chronological calendar age perfectly.
All right, I've got a quick memory trick for you listening to keep this straight.
Let's hear it.
A is for above. Above 0.5 is accelerated aging, and D is for down. Down from 0.5 is decelerated aging.
I like that. Above is accelerated, down is decelerated.
So how did they apply this ADI to actual diseases?
They applied the model to the Zebra Brain Atlas, which is this huge database of single-cell sequencing from patients with different diseases.
And what did they find?
That diseases literally accelerate cellular aging. Brain cells from patients with COVID-19, Alzheimer's, multiple sclerosis, and autism all had significantly higher ADIs compared to healthy controls.
Wait, let's pause there. A respiratory disease like COVID-19 causes brain cells to physically show an older biological age.
It does. A severe systemic disease drastically alters the transcriptome, even far away from the lungs.
Wow.
In the brain, the AI looked at astrocytes and microglia. These are your crucial support and cleanup cells for neurons.
Right.
In a healthy brain, the AI predicts the age of an astrocyte really accurately, a correlation of about 0.84. But in COVID-19 patients, that correlation plummeted to 0.21.
So they basically lost their anchor to biological time.
Exactly. The extreme inflammation threw them into transcriptomic chaos. They stopped doing normal maintenance to fight the fire, and their gene expression warped. They literally aged faster.
That is terrifying.
Yeah. Disease isn't just being sick, it's an active accelerator of aging.
Okay, so if diseases make cells age faster, this brings us to the ultimate paradox. What happens with cancer?
The big one.
Right, because cancer is unchecked, aggressive, cellular growth.
You would assume a deadly tumor is the most biologically degraded, rapidly aging tissue in your body.
Logically, yes. But looking at a pan-cancer dataset, the findings were totally backwards.
Backwards how?
Higher tumor phases like phase 4 melanoma and metastatic tumors like pancreatic cancer that spread to the liver, they actually had lower ADIs.
Down from 0.5 is decelerated.
Wow.
So the AI thinks these deadly mutating cells are biologically younger.
Yes.
But how? How does that make any sense if they're killing you?
Because cancer isn't really about aging, it's about survival and plasticity.
Plasticity, okay, meaning flexibility. Exactly. For a pancreatic tumor cell to break off, travel through your bloodstream, and survive in the liver, it can't act like an old specialized pancreas cell anymore.
Right. You have to adapt.
It undergoes adaptive reprogramming. It returns to a youthful, stem cell-like state, high differentiation potency.
So it's literally hacking the transcriptomic profile of youth to metastasize.
It's weaponizing cellular youth. And we can prove this by looking at normal brain development.
Okay, how so?
In healthy brains, you have these things called OPCs, oligodendrocyte precursor cells.
Precursors, so they haven't matured yet.
Right, they're flexible. Eventually, they mature into fully formed oligodendrocytes, which wrap neurons in myelin. The AI showed that these precursor cells have significantly lower ADIs than the mature ones.
Because potential equals youth to the transcriptomic clock.
Exactly. A precursor hasn't locked into its final job, so its to-do list looks younger. Cancer hijacks this exact mechanism.
Wow.
When they looked at primary pancreatic tumors, they found two groups of cells. One group had higher ADIs. They looked old, stressed, ready to die.
But the other group?
Incredibly low ADIs. They shut down normal functions and turned on genes just for survival, plasticity, and dodging the immune system.
The biologically younger cells are the ones driving the lethal spread.
Yes.
That is just, it reframes everything. scAgeClock isn't just some party tricks to guess your age.
No, not at all.
It proves aging isn't a single uniform process.
Yeah.
But practically speaking, why does this matter for the person listening right now? Where does this go next?
It's going to change clinical trials forever. Until now, if we tested an anti-aging supplement or a new diet or a medication, we couldn't easily prove if it was actually slowing the biological clock at the cellular level.
We were just guessing based on the smoothie.
Exactly. But now we can sequence the single cells before and after an intervention, run them through the feature gate, let the multi-head attention detectives do their thing, and calculate the exact ADI.
We can mathematically prove if a supplement actually decelerated aging in your liver.
Yes. It brings a level of precision to longevity medicine we've never had.
Which is incredible. But it does leave us with a final, really provocative thought to mull over.
Oh, definitely.
If metastatic cancer cells achieve their survival by reverting to biological youth, is the ultimate fountain of youth inherently tied to the risk of cancer?
That is the million-dollar question.
Right. Like, as science pushes toward therapies that actually lower our ADI and bring us back to that transcriptomic peak of age 30, how do we decouple healthy cellular rejuvenation from dangerous, unchecked growth?
Can we turn back the clock without waking up a dormant threat?
Exactly. It's a fine line between rewriting the to-do list and triggering something lethal, something to think about. Keep diving deep.